Claris, one of the hookworm. Other two hookworms are Ankylostomata duodenale and Necator americanus. Necator americanus is called as New World hookworm. New World hookworm. Necator americanus is called as New World hookworm. So, Strongyloides circularis is called as military worm. Was found in Cochin, China in 1876. Human infection mainly caused by Strongyloides stercoralis, and it may be fuel borne. Fuel borne. Adult larvae and egg. Adult worms, female worm seen in small intestine. Habitat, small intestine. Strongyloides circularis. Strongyloides circularis. Only female worm is seen. It is 2 to 3 millimeter long. Male worms free living, present in environment but not human intestine. It lays eggs, oviparous. It lays egg, oviparous. So this is a life cycle. So man and dogs cycle. So man get infected again by Archaria. It penetrates this Archaria and it goes to the lungs and comes back to the intestine, develops into male and female worm. But there is one more cycle inside the human being only. That is autoinfection. Rabidiform larvae in the large intestine become filariform larvae, penetrate the intestinal mucosa for perianal region, perianal skin, and migrate to other organs. So there is one more cycle like this. So this is regular, like other hookworm. Infectiform filariform larva penetrate the skin and of the definitive host. Okay, in the and it filariform larva migrate to various pathways, small intestine. They become adult worms. So parasitic male and female copulate. They lay eggs. Sometimes the eggs are transformed to rabbity falciform larva. Again, go back to the lungs and back to the intestine. So first male and female worm lay eggs. Eggs so becomes rabbitiform larva, filariform larva. Filariform larva are infected to man. So dogs and man, it infects dogs and man. Reservoir host. Okay, symptomatic infection, rashes, recurrent maculopapular rashes. During the skin migration, it can give rise to urticarial rash. It's called as larva currents. Pulmonary symptoms uncommon, occurs only in secondary underlying the chronic obstructive lung disease. So in moderate cases, it can give rise to epigastric pain, diarrhea, constipation, and blood loss. Heavy load of infection gives rise to hyperinfection and disseminated strongyloidosis. Strongyloidosis is very important, one of the opportunistic infection in the HIV. One of the opportunistic infection in the HIV. Stool microscopy, you can see rabbitiform larva. Concentration technique is performed. Yield single stool examination less sensitive due to irregular low output larvae. Repeated stool examination sample is required. So this is rabbitiform larva. This is how it looks. The hookworm and A is hookworm. B is Strongyloides stercoralis. So this is the reference. There's the anal pore, genital primordium, intestine, double bulb esophagus, buccal cavity. It is strong blood vessel. Then up at the top one is synclostomatous. Tento test disseminated strong blood is diagnosed by examining the stool, sputum, and other blood fluid. The tissue biopsy contains number of filiform larvae. Harda Mori filter paper tube method, Behrman funnel technique, agar plate technique, and charcoal culture method. ELISA using crude larval antigen, lip assay, luciferase immunoprecipitation test. Copra antigen detected in the stool, antigen capture ELISA developed by using polyclonal rabbit antiserum raised against Onglotis rati, excretory and secretory antigen. PCR is done. Treatment. Ivermectin 200 milligram per kg for 2 days. Most effective. Albendazole 400 milligram for 3 days. By disseminated strong lordosis, prolonged course of ivermectin 5 to 7 days until the parasites are eradicated. Now, this is a very short this thing about one of the hookworms. Now, I wanted to solve some of the question papers for what it has appeared in the examination. Where do you do Cassinis test? Cassinis test. Where do you do Cassinis test? Cassinis test is done for the diagnosis of hydrated cyst caused by Echinococcus granulosa. Okay, it's an intradermal test. Antigen used is hydrated fluid. Hydrated fluid sterilized by seeds filter. Hydrated fluid used by seeds filter injected intradermally. It's an immediate hypersensitivity reaction. Erythema, redness more than 3 to 6 centimeters, suggestive of presence of hydrated cyst. Hydrated cyst is caused by Echinococcus granulosus. Man get infected by eating food or drinking water contaminated with the feces of the dog, which contains the eggs of Echinococcus granulosus. It gives rise to hydrated cyst in the liver. Hydrated cyst in the liver. This is about intradermal test. Antigen is hydrated fluid sterilized by seeds filter. Separate dextrose agar medium (SDA). It is named after Severods. and it contains dextrose and it contains agar. It's a solid medium and used for fungal growth. Anybody, any idea why it is selected for only fungi? Bacteria will not grow in that. Any idea? Anybody knows? pH is 5.4. Acidic pH. Acidic pH. Acidic pH. That's why it is used as a selective media for fungal growth. Fauna. Now, next question is intermediate host. There are two types of hosts in parasitic disease. One is definite host, another is intermediate host. Definite host where sexual form of parasites reside. Where sexual form of parasite reside. Intermediate host is asexual form of parasite. Asexual form of parasite. Asexual form of parasite. Man acts as intermediate host in two parasitic disease. Man acts as intermediate host in two parasitic disease. One is malaria, second is Echinococcus granulosus. Malaria and Echinococcus granulosus. Draw labeled diagram of influenza virus. It's an enveloped virus. It has got 8 pieces of RNA. It is an enveloped virus. It's an enveloped virus. It's an enveloped virus which has got 8 pieces of RNA. 8 pieces of RNA. On top of that, it has got 2 antigens. This triangular shape is called hemagglutinin. Hemagglutinin. Triangular shaped is hemagglutinin. This mushroom shaped is called as neuraminidase. Mushroom shaped is called neuraminidase. So you have to practice the diagrams of HIV, hepatitis B, and influenza. Rabies virus also. Okay. You have to practice. Written down diagram. Name 3 RNA viruses. One is HIV virus, rhabdovirus, and picornavirus. I'll give you mnemonic for DNA virus, okay? You'll not forget in your life, okay? Triple P, double H. Or sometimes you remember like this. Happy, happy. Or triple P, double H. Herpes, hepadna, adeno, vapova, baro, pox. This is a mnemonic. Or pox, parvo, pepova, herpes, hepadna, adeno. Everybody. Pox, parvo, pepova, herpes, hepadna, adeno. Pox, parvo, pepova, herpes, hepadna, adeno. Pox, parvo, pepova, herpes, hepadna, adeno. Rest are all RNA viruses. You have arboviruses, myxovirus, paramyxoviruses, riovirus, arenavirus. So many are there. Okay, myxo, paramyxo, rhabdo, picorno. Picorno is which causes poliomyelitis. Okay, rhabdo, rabies. Arbo, you have this chikungunya, dengue, and all that. It is the viruses that belongs to the arbo is arthropod borne viruses. Arthropod borne viruses. Now, different species of malarial parasite. Already told you. Make a mnemonic and remember it. Okay. Malaria stands for malair, bad air. Bad air, malaria. So, Plasmodium falciparum. Why it is called falciparum? The gametocyte looks like fox, banana shaped, like this. The fox which is used for cutting the crops, banana shaped. Then Plasmodium vivax, Plasmodium ovens, Plasmodium malariae, and Plasmodium novalace. Novalace. These are the parasites which causes malaria. Which causes malaria. Plasmodium malariae causes fever once in 4 days. It is called as cotton. All three causes once in 3 days. Tertian. It causes every 24 hours. Fever in every 24 hours. No less. No less. That's very important. Okay. Now, how to do laboratory diagnosis of malarial parasite? Very simple. We have You have to do peripheral smear. One is thick smear, another is thin smear, thin smear, thin smear, thick smear for the parasite, and thin smear for identifying species. Thin smear is for the identification of the species of the parasite. Thin smear for the species of the another. Now we will be doing what is called as QBC, quantitative buffer coat. We take the capillary tubes which are coated with acridine orange. Acridine orange, and we centrifuge it and see under fluorescent microscope. They fluoresce under fluorescent microscope. Quantitative buffer coat. Or you can detect an enzyme produced by malarial parasite. One is aldolase, another is histidine-rich protein. So they are like comes like a card, okay? Just like immunochromatography, wax and Pulsive like that. Card comes. So this is a laboratory diagnosis of malarial infection. Now, laboratory diagnosis of HIV. Laboratory diagnosis of HIV. HIV is done by ELISA. ELISA stands for enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. Detecting antibodies against antigen. So you take we usually do indirect ELISA, antigen coated microtiter plate, add serum, and add second immunoglobulin, which is tagged with HRP. HRP stands for horseradish peroxidase. Horseradish peroxidase. So when you add substrate, then the color change. Which can be read by ELISA reader, and it is positive. Okay. The next is Western blot technique. Just the simple, it is nitrocellulose paper in which all antigens P24, P55, then glycoprotein 140, glycoprotein 21. So they are dipped in, just like ELISA, they are put into the paper. You have to get at least 3 bands positive for the. Instead of putting in the well, all the antigens are put on the paper. More precise and confirmatory. Nowadays, we got tri-dot cards. The principle of tri-dot card is immunochromatography. This is the test. and this is the HIV-1. This is the control. This is HIV-1. This is the HIV-2. So there is a lateral. Put the serum and buffer everything, and look for the color change. That gives us an idea whether HIV positive or negative. Next is Cryptococcus neoformans. Remember the classification of fungi as yeast like dimorphic molds. Yeast is like dimorphic and molds. Yeast example is Cryptococcus. Yeast like is Candida. Dimorphic is their yeast at 37 degrees and filamentous at 22 degrees. They show dimorphism. Molds are 5. Rhizopus, Mucar, Aspergillus, Penicillium, and dermatophytes. Dermatophytes. Dermatophytes are again are 3. Trichophyton, Epidermophyton, Microspora. Trichophyton infects all 3: hair, nail, and skin. Tri means 3. Trichophyton, tri means 3. Epidermophyton will not infect the hair. Remember it as Entamoeba histolytica. Microspora will not infect the nail. How you remember this? After M, what comes? Capital letter N. It will not infect the nail. So this is the classification of the morphological classification of the fungi. Remember this. So Cryptococcus, Candida, dimorphic. Dimorphic you can remember as HBC, Histoplasma capsulatum, Blastomycetes, Cocciditis, Paracocciditis. Remember this as HBC. Okay. These are the example of this is the classification of the fungi. Okay. Cryptococcus neoformans is an opportunistic fungal infection. Opportunistic Cryptococcal neoformans is opportunistic fungal infection. Huh? What is? I'll tell you. First you listen to me first. Cryptococcus, they are present in the pigeon's feces. They are present in the feces. And man get infected by inhalation. Man get infected by the inhalation. It either goes to, it is usually gives rise to pneumonia, or it goes to the brain, it gives rise to cryptococcal meningitis. Lab diagnosis is done by doing CSF and doing the capsulated organism. You have to do India ink or nigrosin. Background looks dark and the organism looks hollow. Background looks dark and organism looks hollow. Cryptococcus neoformans. KOH is usually done for dermatophytes, okay, all fungal infections. So, the specimen you get is hair clipping, nails. You have to, with a scalpel, you have to take the scraping from the nail, cut the hair, skin scraping. So then you put it in. Nail is requires 40% of KOH. This is 10. This is 10% of KOH. Put KOH and put the cover slip under microscope. Potassium hydroxide. Nail is 40%, rest is 10%. Okay. Why potassium hydroxide is put is to dissolve skin and keratin material, nail or a thing. So you can see the this filamentous form, fungal elements. We have to report as KOH preparation. Fungal elements are seen. Then you put it onto separate sugar. Look for the growth. Separate dextrose sugar and look for the growth. Then slide culture. What you have to do is you have to take the piece of separate dextrose agar, put Y-shaped glass rod beneath this. A small colony is picked up and put it in the petri dish. Incubate and incubate. After the incubation, the colony will grow on this and see under microscope. This is a slide culture. This is the slide culture. Then microfilaria. Difference between lactophenol cotton blue mount is to stay once you see the fungal elements, you grow on the separate dextrose. Once the colony comes out, then you mount it fungal and make a piece with a teasing needle, a piece, and put the lactophenol bound and put the cover slip. You can appreciate the morphology of fungal elements, whether they are branched, whether non-branched, macroconidia, microconidia, depending on the pencil-shaped macroconidia or leaf-shaped macroconidia, and all can identify the what fungi it is. Lactophenol cotton blue, usually you do it once you get the fungal growth. It is like doing gram staining after you get the growth on the blood agar, Meconic agar, or nutrient agar. KOH is initial. As soon as you receive the sample, you do KOH preparation. Put it on separate dextrose agar. It takes sometimes 3 or 4 days. 1 week it takes. Once you get a growth, then you do a lactophenol growth, and there is a teasing needle. You have to tease the fungal growth and put lactophenol cotton blue mount. And phenol is a preservative. Cotton blue is a stain. Phenol is a, and it is called mounting. Mounting on the slide is called mount. It's a LPCB. Lactophenol. Everybody. Lactophenol cotton blue mount. First you do KOH preparation. Put it on separate dextrose. Then when you get a growth colony, then you do the lactophenol cotton blue mount. Okay. Epstein-Barr virus gives rise to what? Epstein-Barr virus, EB virus, gives rise to what infection? Negri bodies are seen in which infection? Negri bodies. Rabies, intracytoplasmic eosinophilic antibodies. They are inclusion bodies. Name 3 viruses which cause diarrhea. Rotavirus, then Norwalk agent, enterovirus. There's so many viruses. Cyclops acts as an intermediate host in 2 parasitic diseases. Number 1, guinea worm, that is called as Dracunculus medinensis. Dracunculus medinensis. Number 2, Diphyllobothrium datum, that is fish taper. First, Cyclops. The second, it has got 2 intermediate hosts. First is Cyclops, next is fish. Give 3 examples of dimorphic fungi. Histoplasma capsulatum, blastomycetes, coccidioidomycetes, paracoccidioidomycetes. Now, morphology of Entamoeba stylitica, it exists in 2 forms. One is trophozoite, which has got a pseudopodia, which is, and in the ingested RBCs will be there. There is a nucleus, and there will be a quadrinucleate cyst. Quadrinucleate cyst is infective to man. Quadrinucleate cyst is infective to the man. Life cycle, repeat with me, life cycle. Man get infected by consumption of food and water, which consists of quadrinucleate cyst, and they are unaffected by the hydrochloric acid of the stomach, unaffected by the hydrochloric acid of the stomach, and small intestine existation that takes place. And 8 amoebulae are released, and 8 amoebulae get transformed to trophozoite. They produce enzyme called histolysis. They lysis the tissue and gives rise to amoebic dysentery, passing of the stool with blood and mucus. So some of the trophozoites are taken by portal vein, go to the liver, and give rise to liver abscess. From liver it goes to the lung, and give rise to lung abscess. Through blood spread, it goes to brain abscess, subphrenic. It can go to any part of the body and cause a subphrenic abscess. Directly from the intestine go up on the skin, it can cause amoeboma. When conditions become hostile, again cyst, it becomes cyst, and cysts are passed. In India, it's so common that every 1 in 10 or 12 people are amoebic cyst carriers. So we have to eat proper hygienic food, otherwise you get amoebic dysentery. Lab diagnosis, you have to do saline and iodine preparation. Saline preparation, you see the trophozoites, pseudopodia and ingested RBCs. In iodine preparation, you see the cyst, which shows the glycogen mass, chromatid bars, and 4 nuclei. Sometimes pre-cyst is also seen, that is two cyst. Okay, now Ascaris lumbricoides life cycle. I told you other day, life cycle of Ascaris lumbricoides. How man get infected? How man get infected with Ascaris lumbricoides? Which contains embryonated egg, bile stained embryonated egg. Once intestine, they move to the lungs and comes back to the intestine, where develop into male and female worm. Eggs are passed, and eggs development takes place in the soil. Please remember, in the intestine, from intestine they go to the lungs. Okay, soil development takes place, and man get infected. Again, consumption of egg. Okay, don't forget to take it to the lungs and they cause rise to Loeffler's pneumonia in the lung. Loeffler's pneumonia in the lung. So anti-rabies vaccine. What are the anti-rabies vaccine and what is the dosage? Ravi wants to respond. Anti-rabies vaccine, neural vaccine and non-neural vaccine. Neural vaccine nowadays we have stopped because they do rise to post-vaccine. Now available vaccine is Rabipur. Eggs. The prepared in the eggs, yolk sac of egg. Then HDCV. Then Viro cells. These are the 3 vaccines. It is given on 0 day, 3rd day, 7th day, 14th day, and 38th day. 2, 3, 4, 5, 6 injections. That is the dosage form. Okay, anti-rabies vaccine. Life cycle of Pulsive malaria. Mosquito is a definite host. Man is the intermediate host. Man get infected by the bite of mosquito. Sporozoids, they go to the liver, multiply. Merozoites are liberated. Merozoites attack RBCs. Then early ring stage, trophozoite, schizons, and gametocytes in the RBC. Gametocytes are taken by the mosquito and they transformed into zygote, ookinete, oocyst. Oocyst burst and gives rise to sporozoites. Sporozoites infect the man. So first you take to the liver, then come to the RBC. First station is liver. Second station is RBCs. First station is liver. Second station is liver. NIH swab. Where do you use NIH swab? NIH stands for National Institute of Health. Used for sometimes the Enterobase vermicularis is stuck in the perianal region. So that is you have to use. It is a rod, glass rod with the cellophane tape on the top. Okay. ಬರ್ತಾರಂತ hookworm is have to take this. It has got two blastomeres, one meat mud and ಹೇಳು. It has got non-bile stained eggs and it has got two blastomeres. Swine flu is caused by influenza virus only. It has got, I said no, two types of antigens present. These antigens H1, N1, H1, N1, hemagglutinin 1, H3N5 is avian flu, bird flu. H3N5 is avian flu. Arbovirus present in India are dengue, chikungunya, Diasnore forest disease, dengue, chikungunya, Diasnore forest disease, Japanese bay encephalitis. How many? 4. Dengue, chikungunya, Diasnore forest disease, KFD, and Japanese bee encephalitis. Japanese bee encephalitis. Thank you very much.